to act as rapporteurs to note the proceedings of the session. In this session, we have 5 lead talks. One from Dr. M.S. Prasad sir, another one Dr. A.K. Padma Kumari madam, and Dr. T.R. Sharma sir. And then we have a talk from Dr. R.M. Sundaram sir, and the last one will be from Dr. Amresh Chandal sir. With this, now I hand over the session to chair of the session.
Very good morning to all. Distinguished participants, ladies and gentlemen, I would like to welcome you all for this very interesting session on advances in molecular breeding and tools for multiple pest and disease resistance breeding in crops. And today we are dealing with this in this session on rice. You know, rice is one of the most important food security crops, and this year we have become #1 in rice production, producing 150 million tons of rice. But still, you know, we have to overcome many problems which are associated with rice cultivation, and in that context, this is one of the most important sessions which I consider. And also, being working in rice for the past more than 35 years, I also considered it a privilege to be here this morning to chair this session and also to deliver a talk. I would also like to welcome my co-chair, Dr. Viresh Gowda, Head R&D Bayer Bioscience, Hyderabad, and also would like to welcome our former DDG Horticulture, Dr. N. Krishnan Kumar, Dr. Sundaram, Director, Indian Institute of Rice Research, and our very senior colleague, Dr. B.M. Khadi, Dr. Tikku, and all of you to this particular session, and I'm sure you will enjoy the deliberations and discussions during this session. So with these words now, I would like to first invite Dr. M.S. Prasad, Principal Scientist and Senior Plant Pathologist at Indian Institute of Rice Research, Hyderabad, to deliver a talk on disease scenario in rice. In fact, Dr. Prasad was also one of my old collaborator and colleagues with whom we did a lot of work on PI54 and blast resistance during my initial days at IARI. And thank you, Dr. Prasad. You are given 25 minutes to complete your talk. Okay?
Thank
Please
you, sir.
Continue.
Thank you, sir. Respected Dr. T.R. Sharma ji, and then Dr. Gowda sir, and then respected Kumar sir, and then Director Sundaram, and my dear colleagues. The task which I have been given to today's program is that I have to give you a scenario of rice diseases in India. So I'll stick to my presentation to the scenario only. The molecular part and other things Dr. Sundaram will take care of from the rice. So I'll give a brief outline of all the diseases that where they occur, and then can you pass it on the slides? You can pass it on. I'll say next. Just now our sir told that T.R. Sharma sir that India already surpassed China in production. It is the first in the world that we have achieved 142 million tons. And then it is produced in different agro ecosystems. So that all we know very well. Then go to the next slide. Just this slide gives a wonderful how the disease progression took place in rice. Actually, I made 4 breakups like pre-60 and then 65 above, and 65 to 85, 85 to 2005, 2005 onwards. How the agriculture, actually a traditional period to green revolution to intensification in agriculture and then climate change and hybrid era. So how the disease scenario has taken place. In 60s, pre-60s, the disease pressure was low, the two brown spot and blast. And 60 and above is 85, it is a high increase of disease pressure, and then is a blast and BLB again dominated. 85, 2005, high and complex scenarios occurred, and then here the sheath blade has dominated, and then Tungro has taken the toll of rice crop. And then above, after 2005, the very high intensity and the shift in the cultivation, like hybrid rice has come into picture, and then there the blast and false mud has taken over, and then Bakane. Because of this, these two diseases, this is the scenario, the changing scenario over the period of time in rice diseases. This is what I wanted to. In 1945, we know only one disease, that's a brown spot disease, that's a famine, that everybody knows, those who read agriculture. So after that blast, now as on today, that around 13 major diseases are there that are threatening the cultivation of rice. This is the slide that very nicely depicted how the progression took place. Here, what are the reasons that disease scenario is changing or shifting? Here, the main factor is the climatic factor. That as we are all aware that there is a change in the temperature, rise in the night temperature, and also increased humidity due to erratic rainfall. Rainfalls are very erratic, and frequent floods and droughts, and extended cropping windows, like the cropping window is extended, and then that favors the survival of the pathogen. These are some of the factors from the another factors are changing in cropping systems. Like now we are advocating the farmers that we go for the DSR mode of cultivation. There the disease scenario is differs, that brown spot again it is coming up very high. So in some places the irrigation actually very high intensive cultivation they are doing, like Telangana I took as a model, that in recent times in Telangana the rice cultivation has gone up very high. So there the pathogen system also, disease also very high intensity throughout the year. Like continuous cropping, like season by season we are going only rice. These are some of the factors that contributes in the changing the disease scenario. And as we very well know that the host genetic factors, like only few popular varieties that are specific to some regions, like in Andhra we go only BPT, in some Tamil Nadu Ponni, like there several varieties are there. So those farmers they prefer those varieties only because of their quality and all, but they are susceptible to the pathogens. So that breakdown of the resistance is another problem. Now at the pathogen front also there are changes, pathotypes, new pathotypes and new races are coming, and then adaptability, and then virulence, increased virulence, and survival during off season also increased. Then another one more factor we have to take into consideration. Now use of the N factor is very high, but people are not bothering about how K is to be utilized, and then micronutrient management also people are not bothering. That's why some of the diseases like blast and BLB, they are very high incidence had been happened in recent times. Now this is the actually picture what we generally see in rice, that in regarding fungal diseases there are around 13 diseases are there. And then bacterial diseases, generally two are made dominant, one is BLB. In viral diseases, we know only Tungro, but now recent times, one disease, the southern rice black streak dwarf virus are known that in Punjab and Haryana, how it is threatened cultivation of basmati. You take the example of Bakane also. Bakane also recent times, the increase has been very high in Punjab and these places, Haryana, where the basmati rice is being cultivated. False mud, as you all very well aware that it was once a minor disease and then it is a Lakshmi disease we call, but now a stage has come without managing the false disease in hybrids. I think that cultivation is unsuccessful and they are getting a lot of losses. The other diseases like blast, every year it occurs. It takes, even still it is a major disease, and then brown spot now it is aggravating year by year, and then because as I told earlier also that the DSR shifting of the cultivation, and then people are taking in upper lands, and then where the nutrient management is not proper, this disease is very much aggravated. Leaf scald, this western belt of India, the leaf scald is very much Konkan region and all. Leaf scald is also very severely occurring, and the sheath blight, once it is known to occur only in the coastal belts of West Bengal to down to Tamil Nadu, but now it has gone into other parts of the country, like in Gangetic plains and then Punjab, Haryana, even in North Indian also. You will be seeing in coming slides. Udavata disease, earlier known only in Karnataka, now in few pockets this disease is also being observed. And then stag-born, and then bunt, narrow browns. But these are all now, they are the minor ones, but we don't know what a scenario will take place tomorrow because of these outbreaks. Actually, the strength of our IARIs, we have around 48 funded centers. There our group is State Agriculture University people are there. And then we generally work on host plant resistance. That again 6 diseases every year we screen them, and then we take them a disease resistant lines, and then we give it to the breeders, and then this is our general program. And then virulence also we will monitor, like 2 pathogens, blast and BLB. And then management aspects, we take care of the management aspect of all these fungal diseases, like using fungicides. Nowadays only fungicides we are not, site fungicides we are not using, we are using the mixed fungicides. And then now one more, here in this context, like disease scenario, POS only plays a major, because this is also one aspect we take care of at IARR, this production oriented survey. Our team, that plant pathology team, along with other groups like breeders and then agronomists, they go out for the survey during the crop season. In the entire 48 centers, like it spread across the country, as you have seen. Then they survey it, what are the diseases are there in that season. 2 to 3 times they go for survey, and then they analyze the data, and then they'll send to us what are the, now which is the disease happening more in which place. Like now this happens in this disease scenario aspect, production oriented survey, I take as a background, and then we'll discuss about each disease, how the change has happened. So our team actually comprises of all agriculture department, line department people, and the State Department of Agriculture, and also our university staff, and also our ICR employees. And then we surveyed almost all the 125 districts every year. And then we put forward that these are the 5 diseases like blast, BLB, seed blight, false smut, and brown spot. They are the major 5 dominant diseases that are threatening to the cultivation of rice. Now blast, as you know very well, that is a leaf and then node and neck blast. How the leaf blast is also very tricky. Earlier it happens to occur at few places, because now it has gone to several interior places where earlier it was not reported also, because of change in the climate or because of wind. The wind what we are using it that for crop period, that is also very less now. As I told that this is a production oriented survey. Every year we have a data of a repository for the last 30 years. So we have analyzed the data of the 30 years data for all the 5 diseases, and then we come out a map, that the decadal map of each disease, and then that map shows that how disease has progression has happened. Now I will present for each disease. Oh, this is not clear. Then one, I think you missed. It's not clear. Okay, I'll explain that. You just go back. Oh, it is not coming. You just, yeah. Wait. Oh, it is. I think something happened, sir. I will proceed further. Yeah, I have. मैं आगे जाता हूँ. Sir, leaf blast, it is also in the 3 decades we have taken the maps of leaf blast. It has shifted from 10 years to 10 years. Now it has gone to the deeper into the different places. Now this is a case of neck blast. Don't do it anything. I'll do from here. You just stop that window also. Yeah. Okay. This is a neck blast scenario. Earlier neck blast was not that much prominent. Now these increased rain during the flowering and ripening stage, mild night temperature increase, and then high use of N application, and then only one variety, that's what I told, the preferential variety that is available on the ground, that favors the enhancement of this disease, very severe levels. In 81 to 90, this was very sparse in the country, you see the dots, and then in 91 to 2000, it was a little bit aggravated. Now you see the present scenario, it has gone to the, wherever rice is grown, I think now neck blast is also a major problem. Actually, at DRR, we have regarding this topic is actually related to genes and then molecular level. So I wanted to show some of the work what we did at IARI in collaboration with Dr. T.R. Sharma sir. Sir has told that we have several common projects were there. So in BLAST, we have taken the first work at IARR in collaboration with IARI. Dr. Sir was the main PA for the time. And then we didn't have that monogenic lines background, in the background of different genes what we have, like PA1, PA2, PA9 and PA54. These are the 4 major genes available. So we didn't have that monogenic lines. For BLB we have monogenic lines, but here we didn't have. So our idea was to develop monogenic lines, a project we were closely associated, and then we developed monogenic lines in the 2 backgrounds, 2 popular varieties like Swarna and then BBT. So different lines, why these lines we developed, I will tell you, again it will come. So in those 12 lines what we have developed in the both the backgrounds, 2 out yielded the other lines that were, so the 2 were released as a dearer than 51 in the background of which is having a PA2 in the background of Swarna, and then dearer than 62 it is having both BLB and blast resistance. So those monogenic lines, why we developed, because we wanted to give as a donor to the directly used for breeders, but in that we have put those as suggested by Sharma sir during that time as a DDG. We took these aspects, we put them in the 48 locations, how the genes are performing. So we are given a gene map that in zone 1 what are the genes that are really working well for blast, and zone 2 and zone 3, zone 4 up to zone 7. These are the different combinations and the different gene combinations we have given. Out of all, the PA54 was the best one in most of the zones. And now we discuss with the seed blight. Actually this seed blight disease is easy to work but difficult to manage, because it grows very well in the petri plate, but very difficult to manage this disease. Once it comes into the main field, I think years together it will be there. The sclerotial body survive, I think 6 to 7 years. So here, this is a picture in the global level how this disease is there. And then one of my scientists, Dr. Prakasham, is working on this disease along with Dr. Sundaram. And then here also we made some maps. Earlier this disease was known to occur only in the coastal belts. But now you see the recent map, seed blight, it has gone to very high level in the northeastern and then Gangetic plains, and also in Punjab and Ludhiana. Very high intensity, because here the challenging task is there, we don't have any source of resistance. There in blast we have different source of resistance, but here we don't have a source of resistance. At IARR, we have identified some source of resistance like some QTLs in the Tetap, and then in the background in Tetap, and also Vajupek. I think Sundaram will tell about in detail about those genes and then how they work in sheath blight. Bacterial blight. This is the actually disease. Earlier it was very minor, very mildly it is appearing, but in recent times, I think it is appearing in a very severe form. Wherever you go, I think it is threatening the rice cultivation. Dr. Laha, one of my colleagues, is working on the disease, and then with the help of Sir Sundaram, both together they work. This is a bacterial blight, bacterial leaf streak, and panicle blight. These 3 are present, as rest 2 are, I reported, they are not in India. Again, some problem has come. Okay, this is also the map which he missed actually in the how it time series it changed the area to that area to, how intense this disease was. This is international. This is a global picture of BB, mainly in Asia and some parts of Western Africa, and also USA and then South America. These are existing in those places. In India, it is endemic, mostly in Punjab, Haryana, Uttar Pradesh, parts of Uttarakhand, Bihar, Jharkhand, Chhattisgarh, West Bengal. Most of the rice growing places it is there, as you see in the map. Because this is a major, now it has become a major production constraint. Here we can't manage this disease by chemicals, because fungal disease there are chemicals available. At least you can manage if you take rightly application right time. But here this disease is not having, this is a bacterial disease, we don't have chemicals for this disease. We have to go through certainly through host plant resistance. Very great work has been taken up at IARR with Dr. Sundaram and team, and then in that Dr. Laha is one of the person, and we all took forward this work. This is a map, decadal map of this bacterial blight. This is also very severe form. In southern India, it increased a lot, like Kerala, Tamil Nadu, Andhra, Telangana. These incidents nowadays is hampering the cultivation. Even in Punjab, Ludhiana also this disease is very severe. These are the things that in some pockets it has gone down, in some pockets it has increased. Like Punjab and then Haryana, that belt, it has come down a little bit over a period of time, little bit. But here in southern belt, it's aggravated like anything, like Andhra, Telangana, Karnataka, Kerala, Tamil Nadu, Pondicherry. This you see the red map here, it is the most, I think, very severely it is occurring year by year. This is the intensity you can see, bacterial leaf blight, how it is there in East and West Godavari districts of Andhra Pradesh. This is in Telangana. You can see acres and acres it has gone like that only. Here the main, actually the intervention came from IARI, the first variety, that is a gene pyramided line developed by Dr. Sundaram and our team, and there is improved Sama Masuri. It has taken a solace and then it has gone into deeper into the farmers. Now that cultivation has picked up. Again, that also a little bit toned down, and then again we identified a gene 38 and then XA 38, and that has been incorporated. That is taken still further strength. Still some more gene, one more gene they identified. They are putting it in the background. This is dearer than 53. It is a wonderful variety. And then you see the incidence of BBT and all dearer than 53 in Telangana. These are some of the lines which are having bacterial leaf blight resistant genes. These are the nutshell about bacterial blight. And then one of our colleague, the Basavaraj, is working on brown spot disease. I told you that this disease is picking up very fast, and then because of changing in the cultivation pattern like DSR and all the methods, we know only two famines in the Bengal famine, and then 1918 is this Krishna and Godavari Delta famine. But now, this is the scenario of this disease. This disease is there in mostly Madhya Pradesh, Uttar Pradesh, Bihar, Chhattisgarh, Odisha, Assam, Jharkhand, Punjab and Karnataka, AP, Telangana, West Bengal. In a very severe form nowadays, this disease is there. You can see in these maps, in decadal maps, how it is progressed to after 10 years to 10 years. In some areas, what are the real, in case of Chhattisgarh and Jharkhand, he has gone further and then identified the pockets of the places where it is really affecting the cultivation. This is, see the severity in the survey you have collected such a severe symptoms are there. Here the one work I would like to stress here, this is actually very sparse growth, but maintaining one culture itself is very difficult, brown spot. But here at IIRR, we have developed a wonderful, that Basavaraj, and then we have developed a wonderful technique to sporulate it. Actually, sporulation is a major problem here. So he has developed one technique with a rapid food agar medium. He is going to patent it, and then using this technique in art, because we need to screen a number of lines in a short period of time. This is a facility we have created. When Dr. Sir was the DDG, T.R. Sharma, he has inaugurated this facility at IARR. And then these are some of the work has gone further to identify some of the resistant lines in the brown spot, and then identified few resistant lines, and then they are being now put it into the breeding program. Now another disease, this is one of the false mud disease. Lata Lakshmi is working on this. Since she joined, we asked her to work on this disease only, because that time the etiology was not known. Even fungus also, it was not even culturable properly. Now we have gone to a level that we have a repository of, I think, 200 to 300 lines we have, cultures we have across the country. And also this disease, artificially we have inoculated. Artificially we have created the disease. In this is maps I wanted to show this severity. Here also the false mud, after the advent of hybridizers, it has gone up like anything in the 2013, 23 years. You see the change in the disease pattern. And now at IARR, we have developed a facility called artificial screening in a large scale germplasm of rice. This is a wonderful technique, and it has been applied for patent. In field evaluation also we have gone. This is a field evaluation technique. With hypodermic system they inject, and then typical type of the symptoms it will produce there also. It's a wonderful technique, and then it has been, patent has been applied. And the strength of our institute is actually molecular breeding people are sitting here. And then if you want to be a successful molecular breeder or molecular person, you need to have thorough phenotyping background work like pathologist. So we can clearly say that we have for blast wonderful screening facility UBN, we have false mud screening facility at IARR, and also sheath blight, bacterial leaf blight, brown spot. Now we have gone for sheath rot. There's one Jesudas who has joined and he has developed wonderful technique for sheath rot and also rice tungro. I think tungro, nowhere that screening facility is available, I think except IARR, I feel. So it's the only place where we are getting it screened. With this, I think with 1 or 2 slides, I'll project this management of rice diseases. You know very well that is a host plant resistance is a major what we are looking at IARR, and then several lines have been developed. And then now one scientist joined on biocontrol aspect also, very good work he has conducted. Anyway, the fungicides, fungal diseases without fungicides, when it is in severe form, we have to go for resort to the fungicide supply. Maybe now mixtures of fungicides we are doing. These are some of the lines developed at DRR using with the different genes, gene pyramid lines. Is a collaborative work of breeding, crop protection, and also biotechnology. This I told you that one person joined is a Kannan. He has identified 3. One is Trichoderma aspergillum, tyke, and then bacillus species, and also one more is there, Pseudomonas species. These 3 he has now gone to another level of delivery system, and then he has about to patent the mechanism of delivery and everything. It's wonderful work, and these are all maintained against fungal diseases. Their utility was there. Slide has gone. One more slide was there. It was IPM slide, sir. Without IPM, I think we have wonderful 7 zones are there, rice zone. At each zone, specific IPM module has been developed. So with this, I thank all of you for patiently listening. Our chairman, sir, and my director. Without their help, I think it will be my colleagues at IARI, their help. Otherwise, it is not possible to do the work, sir. Thank you, sir.
Thank you, Dr. Prasad. Excellent presentation, and you have given us an overall scenario of all these diseases, how these are becoming prevalent now, which have become obscure in previous years. So that's really very good, and I don't know whether we can take questions. We have time for question, 1 or 2 questions. Yeah. He is asking about
I got his point. Rice tungro. Tungro actually in recent times, our survey says that its incidence is very minimal. That too, it is in small pockets in Tamil Nadu and then in West Bengal. It is there, very small area, and the intensity is also very low. That's why the scientists are also not coming forward to screen it. Yes. That's why we had only, because since it is a rice institute, we cannot ignore that screening of the disease. We are doing at IARR level only. But as per the intensity level, it is very less. One disease I told you that southern, that is there now, but it is also very erratic. 1 year it was very high on basmati, and next year it disappeared. Again, last year a little bit was there.
One more, sir.
Yeah.
I seen their screening protocol, their neck blast screening protocol not there. So you are working on
because I don't want to, because neck blast protocol is the protocol we have developed. That is also one of the, because neck blast screening is very difficult. Leaf blast, okay. Yeah. It is a standardized protocols. But neck blast we have developed it IARR, that I cut the slides because of time, paucity of the time. It was there still last night.
Maybe you can discuss later.
Yes, you can discuss. Because a wonderful protocol is there for that.
Yes. Thank you, sir.
Thank you.
Good morning, sir. Just I have to know clarity. When we see the severity of leaf blast and neck blast, but in the field there will be less severity. Otherwise, no disease in the leaf blast at early stage. But same field, when we see at the panicle stage, we will get very severe of neck blast. What will be the reason?
Actually, I told that blast appears because that 3 critical growth stages are there for blast. One is leaf, another is node, another is panicle. So at three critical stages. Why I am telling critical stages? On these critical stages, the management aspect depends. So when in these critical stages, the climate, because blast is very much prone to the climatic factors. That conducive climate is available on and then a susceptible variety available on the ground, it will knock. Suppose it may be the escape at leaf blast, and at neck blast, these conducive conditions are prevailed. That's why the disease has been very rampant. Nowadays, the season is little bit progressed. When the neck blast, in November and this earlier, the season was a little bit earlier. Now the season little bit more. That it extended to November. That panicle emergence. That time either humidity or wind splash of some splash rain, they are there. That is favoring the disease development. That is the reason why.
And one more, sir. How about the PI2 and PI54? Is it working all over, uniform working?
PI54 is the, I told you also, because SAR is there, I am not telling, but the PI54 is very strong, because it had the capacity to knock down almost all the pathotypes.
All over
the country. PI54 had that, even in hilly area, plains, everywhere. PI2, PI1, they had limited this thing.
Maybe last question from नहीं आपका हो गया.
Okay.
Is it that PI54 is means resistant for neck blast or what?
Yes, sir. Yes, sir. It is there.
Yeah.
It's there.
Is it?
Okay, thank you,
Thank
Dr.
you, sir.
Prasad.
Thank you.
We will stop this discussion now. Thank you for excellent presentation.
The main questions, whatever that they have, in case they still have questions, they can bring it to the him or you.
Okay.
Then that,
Okay, but we are getting suggestion from the organizer that those who have not able to ask question, they can send by mail all those questions to Dr. Prasad or to Dr. Meenakshi Sundaram or to me, because then we can reply. Yeah. Thank you very much. For next presenter, I will request my co-chair to invite him.
Thank you. Now I would like to invite Dr. A.P. Padma Kumari, Head of Entomology, IIR Hyderabad, to talk on current insect pest scenario in rice. It will be online.
Unfortunately, the screening protocol was not available earlier, but now they have to. Yeah, very. Have you seen the? You should go only. The Kani is coming in a big way now. Even Pakani and Jagan are.
She is speaking and collaborating with him. Hello? No, we can't hear you, madam. It should be okay now.
Am I audible?
Yeah, now we can hear you, madam. Please go ahead.
Oh. You are sharing the slides? Are you sharing the slides?
Yeah, our team is doing that one, madam. Just give us a minute.
Yeah. Okay. Or I can share from my end also if you permit me.
Yeah, that would be good, Dr. Padma Kumari. Please go ahead. Yeah.
If the host can permit me to share the slides, I can share. It says host is not allowing sharing for this meeting. So host has to allow me to share the slides. No, meeting ID is 875-9478-7484. Meeting ID. Are you able to see me? This is, the meeting ID is 875-9478-7484. And the passcode is 58005.
This meeting is being recorded.
875-9478.
Yes, we should have.
I think slides.
Yeah, we can see your slides, madam. Please go ahead.
Voice audible to you all?
Yeah.
Okay, thank you. So, very good morning to you all. I'll start my presentation. Respected chair, co-chair, dignitaries on the dais and off the dais, faculty and students. Today, I'll be speaking to you on the current status of insect pest scenario on rice. And we can see that rice is grown in many ecosystems, like it is grown in many ecosystems like direct seeded rice, hilly rice, Kuttanad area, irrigated rice and deep water rices. And all these cultivation systems influence the pest scenario. And if you go into, I'll be briefly talking about the pests of rice. And there are 100 insect pests that inflict rice, but 7 are important. And there are some regional pests confined to few regions, of which 5 are important. And we can see that 5 are. And here in emerging pests, there are whiteflies, rice mealybug, termites, and panicle mites that are of growing concern. And if you see the importance of the rice pest stages and the insects that inflict the crop, most of them are vegetative feeders. And in the early stages, if we take chemical control appropriately, the damage is compensated in the vegetative phase. But there are some insects where the tiller damage is like gall midge or stem borer and BPH, which affect from right from sowing till harvest. And here lots of compensation is not possible, and invariably we have to look for other mechanisms of tolerance. And most of the, if you look at the stem borers, gall midge, leaf folder, plant hoppers and gandhibug, these are the major pests of national significance. And in stem borer complex, there are many species, of which if you see I can say that yellow stem borer is the most dominant pest among all species of stem borers, that is the Syrpophaga insectivus. But there is white stem borer in the Himachal area and in the Kuttanad area, which is majorly a vegetative pest in Kutanad area, but white stem borer in Himachal, it causes both vegetative and reproductive stage damage. But of late, with the change in cultivation practices, you can see that it is gaining pink stem borer and dark-headed borers are gaining prominence. And here, if you look at the damage, everything is dead heart or white ear. And here we can see that across India, it is almost above ETL. And white ear damage, it is spread across India, it is again above ETL. This is the average of 5 years data from our ICRISAT reports. It is spread widely across India. But when you look at the species composition, you can see some are yellow stem borer, which goes deep into the root system and it survives in the stubbles. Pink stem borer is gregarious and it causes lots of tunneling. And dark-headed borer, it causes both white ear and dead hearts, and it is gregarious in nature. So the damage they inflict is much more as compared to yellow stem borer because there will be extensive tunneling in the tillers. But we only worked ETLs and 10% dead heart is taken as a criteria, and we have 6 pheromones only for yellow stem borer control. Coming to the species composition, in one study in Srikakulam area and West Godavari and Nellore of Andhra Pradesh, in rice pest cropping system, we could see only yellow stem borer. But wherever there was the rice maize cropping system, we could see in West Godavari pink stem borer followed by yellow stem borer. But when there were other cropping systems adjacent to rice like sugarcane, ragi and other millets, we could see 3 species that are occurring in the rice crop. It is mostly dark-headed striped borer, pink stem borer and yellow stem borer. So the cropping systems also do matter in the occurrence of the stem borer, but mostly all the symptoms are either dead heart or white ear. And in deep water rices, it's exclusively by yellow stem borer where it can survive and flourish. And coming to the gallmage, here you can see in the coastal...
The recording has stopped.
All through you can see. Am I audible, please?
Yes.
Am I audible?
Yes, madam.
Hello?
We can hear you, madam. Please go
Am I
ahead.
audible? Can someone tell me whether... Am I audible?
Yes. We can hear you, madam. Please go ahead.
Thank you. Because recording has stopped, then I wanted to confirm. So here you can see that in the gallmage here, it is along the coastal belt and up to interior Sakoli we can see, and it is always above the ETL. And here you can see that the tillers are damaged, and if the variety is having more tillers, there will be a bit of compensation, otherwise the losses are huge. And even the stubbles, they serve as a source of inoculum, and ratoon rices, they are a big source of inoculum. Here you can see that the feeding stage is very less of the maggot, and people go in for sprays. As the feeding stage has a low duration, spraying is not effective. We advocate granular insecticides, and of late not many granular insecticides for this general purpose are available. And this BPH you can see, it is a dangerous pest and it keeps sucking the sap from the tillers and it debilitates the plant, leading to hopper burns. And here you can see the whole tillers, they get chappy, and then it travels from plant to plant and becomes what you call, it loses its vigor. But coupled with unseasonal rains and lots of infestation due to migratory population, most of the lodging occurs at this particular stage. And this is always above ETL in most of the areas, and most of the insecticides are directed either to BPH or to yellow stem borer. And here, due to unseasonal rains, we can see that lots of damage is occurring and people are not able to harvest the crop because of lodging. And coming to whiteback plant hopper, most of the time BPH and WBPH coexist, But in here, WBPH actually colonizes the crop, and after that, BPH takes over due to its high multiplication rate. But since 2022, we could see that some of the states, this southern rice blackstreak dwarf virus, also known as Fiji virus, is gaining prominence. And what we could see that due to the spread of this virus, the infected plants develop narrow upright leaves, weak roots. And here you can see that PR131 is the highly susceptible variety. And farmers are uprooting the plants, and the cost involved is around 10,000 to 15,000 per acre on resowing, and about 15,000 per acre for insecticides. And not many farmers are willing to take up rice because of this particular virus, and close monitoring is essential in the nurseries to prevent the spread of the virus. And the next one is the GLH, that is the green leaf hopper, where this year, after a gap of I think 5 to 6 years, we could see tumoro infestation in the West Bengal area of Chinsura, and it's a xylem feeler, and nearly more than 50 acres has been damaged by this particular pest, causing tumoro-like symptoms. And here we can see that virulence monitoring is a constant endeavor in our studies, and we could see that we monitor virulence for brown plant hopper and galmich through a set of host plant differentials. And we could see that our Indian populations vary in their reaction to certain genotypes. So we should have our own donors in conferring resistance to this particular BPH or to galmich. And it is a vicious cycle. Once you deploy a resistant cultivar, there would be virulence, and once virulence is there, again we look for more genotypes, and we are going for pyramiding of genes. I think GM1 and GM4, or GM4 plus GM8 and GM1 plus GM8 are more effective. Coming to the sources of resistance, land races and breeding lines are being exploited. Once we don't find any significance in these significant donors in these lines, we look for wild races and mutagenesis. In such one endeavor, we could find that through EMS mutagenesis, we could identify few sources of resistance for stem borer. It can be called as tolerance at vegetative phase. And out of for BPH 51 genes identified across world, only 4 to 5 sources are working very well for Indian populations. WBPH genes have to be exploited. Galmich genes, 11 genes we are deploying, out of which only 3 genes are very effective, and we are going for gene combinations. We have identified leaf folder QTLs and YSB QTLs. This knowledge is very much useful for this symposium, where we are looking for molecular breeding tools. We have donors for both the insect pests, multiple insect pests, and donors conferring resistance to both galmich and BLB, and heat tolerance and WBPH in N22 mutants. Leaf folder, this is also a major pest, and you can see it is spreading across India, and it is always above ETL. And there is a complex in this, but of this, Nephrocrosis is the most dominant species. And here you can see that in the early stages, just after transplanting, if there is a leaf folder incidence, there will be less possibility of recovery. So insecticides, there is a must. But losses will occur again in the flag, if the flag leaf is affected, because chlorophyll in damage will be depleted, and there is reduced turgor. And this causes losses in the yield. Definitely it will have loss in the yield. And this species composition is important, because when we are deploying pheromones which are biorational approaches, we should know which species we are addressing to. So more detailed studies on the species composition at very location specific is important. And most of the insecticides are directed towards leaf folder, because after a certain stage in the crop, they start moving in large numbers to another green crop. And farmer always thinks that these are migrating and they cause lots of damage, and they tend to spray many insecticides. Earhead bug or gandhi bug, it causes pecky rice, and it also affects the quality of the grain. And here also species is very much important, but all of them cause pecky rice. Thrips are gaining prominence because in the nurseries where there is a drought for short spell of drought for 10 to 15 days, you can see the tips of the leaves being rolled out, and this causes drying. There is resistance to this, but appropriate chemical control will address the problem. And you can see in some parts this is a common thing. Insect pests of regional significance, you have termites, white grubs, swarming caterpillars, grasshoppers, caseworm, cutworm, hispa, and mealybug. And if you have hispa here, it is more in the hilly tracts of Himachal and Assam. It scrapes both grub and adult, they cause damage. It scrapes the chlorophyll. And if the vegetative phase it occurs, small seedlings, if it affects the small seedlings, recovery would be difficult. But in the mid-tillering phase, if there is any infestation, definitely it would be recovered. And insecticides is the best possible control measure. And here in caseworm, in Monkombu, Brahmawar, Karaikal and Rajnagar, this has become a serious problem because if there is drainage problems, lack of drainage is increasing this pest, and of late this is gaining prominence and it is creating vegetative phase damage, and again insecticide or pulling a rope will be the alternates for this control. And grasshoppers are a perennial problem in Kudwani area of Jammu Kashmir, and it is also observed in Adutara area, and white grubs are a problem in Kumaon region of Uttaranchal. Emerging insect pests are mites, which will be leaf mite, which is always there, but panicle mite is gaining prominence. Blackbug, blue beetles, and whitefly in rice. Here we could see that blackbug outbreak was reported in 14 when there was an unseasonal rain. In light trap catches it was there up to 23,190 in 2 days. These unseasonal incidents of rains, they create a favorable environment for this pest to come onto the rice crop. The symptoms of damage are they suck like a BPH and debilitate the tillers, but they also cause white ears, so it is all the more dangerous. But it is restricted to Aduthurai area of Tamil Nadu. And here we could see that appropriate monitoring and insecticide measures will take care of this particular insect. Hall maggot is gaining prominence, and you can see all along the coastline it is almost previously it was confined to seedling stage, but now panicle initiation stage also we are seeing this insect. There are few doubts about the species that is causing incidents in the panicle initiation stage because there are losses are high. We are yet to confirm whether it is Hydrelia species or any other different pest, and the work is underway. In rice-based cropping systems, wherever it is rice, we could see that stubbles, they serve as a source of inoculum for both the yellow stem borer larvae or galmage or BPHX. And winter nurseries, their source of inoculum will be the infected nurseries. I think last year nearly 1.5 lakh hectares of area was infected in the Tungabhadra command area by galmage, and the source of inoculum was spread through infected nurseries. And harvesting through mechanized harvesting, lots of stubbles are left over, and these are the source of inoculum. And weeds are the main sources of inoculum for all these insect pests, and we have to take care of them. And armyworms and cutworms, we can see they just are sporadic in nature. They come, as the name indicates, they come in huge masses and they devour the plants. So only chemical control is the best option. And we have to be vigilant about how the things are going about. And it was found in Mandya district, Chhattisgarh, Odisha, Telangana. And in Maitimna also we could see in Anantnag and Kulgam districts some time back. And it is also reported from Jagdalpur, Patambi and Warangal devouring the whole crop. overnight. And a new pest was reported from Undi area of West Godavari, and this was attributed to the spraying of bifenthrin in rice. Frequent application of synthetic pyrethroid has increased the incidence of Bemisia formosana, and which had led to yellowing, sooty mold formation, chaffiness, and there was finally drying and twisting of the panicles. This information was shared by the in charge from KVK Undi, Dr. Mallikarjuna. And paddy leaf mite, yellow mite is always there, and wherever there is a short spell of drought, we can see that, and one rain would take care of it. But panicle mite and sheath mite is taking the toll these days. Here we could see that there will be black lesions on the midrib. People ignore it. If they take a prophylactic measure, that would take care of it. But here people just ignore it, and once there is a rain and the panicle starts emerging out, we could see that most of the grains are sterile. And it is always confused with the dirty panicle. And we could see that most of the farmers think that it is a disease and they go for fungicides. So still people are not aware about or not clear about the dirty panicle that is caused by mites or due to fungus, and there should be an awareness campaign for this. Bloodworms are picking up now. Since 3 to 4 years, these are reported from Punjab, Gujarat, and this year from Chhattisgarh and Nellore also. And in direct seeded rice areas, you can see these red color worms. They are also called as red worms. And there are few studies which say that they are feeding on the fresh foods. So a further etiology has to be studied. Now SRA, system of rice cultivation, then DSR versus pest incidence. Wider spacing adopted in SRA cultivation may favor increased hispa but reduce garbage. In the tillering stage, vigorous plant growth with clusters of tillers may attract defoliators such as cutworm, ear-cutting caterpillars, and leaf folder. However, significant increase in number of tillers and leaves should be able to sufficiently compensate the loss due to defoliation in case of SRA. And SRA cultivation may reduce BPH incidence due to increased aeration. But in some areas where DSR is practiced, when the spacing is not taken into account by the labor who do close-up planting, there is an increase of BPH incidence that we have to take into account. Now, nematodes are gaining prominence, and with the change in system of intensification, system of cultivation, DSR, SRA, and aerobic systems, there is an outbreak of Maladogen graminicola. Many years ago it was there. It was in the nursery. Now it is coming into the main field also. And this year also in Mandya area, we could get the reports that Maladogen is there. So it is everywhere now. White-tip nematode is confined, is seed-borne and has quarantine significance, and it is prevalent in humid areas. Ufra nematode is common in deep water rice. Rice root nematode occurs in irrigated and semi-deep water rice. Root lesion pratylincus are serious problem in upland and aerobic rice. Now, effect of organic manure, it is more effective when we apply organic manure, that is neem cake and poultry manure, which reduces stem borer as well as rice root nematode populations. Vermicompost application can also result in low nematode populations. But these organic packages are more adopted in basmati rice, but can we really cater to the need? Can we really control if the pest is beyond epidemic proportions? And that is a big question mark. Now coming to the toxicity of the
Dr. Padma Kumari.
Insecticides, what we found is with the ban of insecticides, not many granular formulations are available.
Yeah.
And
Dr. Padma Kumari, it would be nice if you can close the presentation in next 2 minutes.
Sure. 2 minutes, sir.
Yeah.
So dealers invariably use and they control the market. State policy and availability of the insecticides matter. Biopesticides 2 through 3 sprays are effective. And finally, with the change in pest scenario, we have 3 to almost 21, 23 pests now. and each has their own role in causing damage. So a basket of options are available for rice APM, but we can address DPH, WBP, and Stembora for host plant resistance. And we have a few mobile clinical labs where the knowledge will be shared in various languages to the farmers. And future researchable issues are pyramiding of insect-resistant genes in allied background, location-specific IPM modules, which is a must, exploitation of susceptible genes, secondary metabolites and metabolic pathways, VOCs, HIPV and HAMP for pest surveillance, habitat management, utilization of food cues for conservation biology, and exploring gene editing as tool. Finally, my word is know your pest, that is KYP, and know your ecosystem, so that pest surveillance and monitoring will add to it, and we can identify the dynamics of the pest. And emphasis on nutrient management, behavior, monitoring of virulence is a must. Location-specific deployment of varieties with a durable broad-spectrum resistance with good quality traits is the need of our. We can look into induced resistance and cardamons, and sensitization of location-specific IPM to the agricultural officers. Thank you. I thank our director and the organizers for facilitating this Zoom meeting so that I can share my thoughts with you. My colleagues, Dr. Chitrashankar, Padmavati, Sridhar, Raghavendra, Somsekar, and Chavan, who have given their inputs. Thank you one and all for your patient hearing.
Yeah, thank you so much, Dr. Padma Kumari, for giving an excellent overview on insect pest